Hi, and welcome to my presentation on the digestive system. So first we'll go over the organs of the digestive system, then its functions. You can divide the organs in the digestive system into two groups, the accessory organs and the organs of the gastrointestinal tract, which is abbreviated the GI tract, and it's also called the alimentary canal. So the GI tract organs are the mouth, which the proper term for it is the oral cavity, then the pharynx, the esophagus, the stomach, the small intestine, and the large intestine. And those are all in order from the beginning of the GI tract to the end of it. Then the accessory organs are the teeth, the tongue, the salivary glands, the liver, the gallbladder, and the pancreas. Those all impact the food in one way or another as it's moving through the GI tract. So over here you have a diagram of all of that. Here you have the oral cavity, then the pharynx is the throat, then the esophagus goes down to the stomach, then the small intestine is the bulk of the intestine, and the large intestine leads out into the anal canal. For the accessory organs, the teeth, the tongue, the salivary glands, those are all around the oral cavity. Then the others are in the abdomen. You have the liver next to the stomach, the gallbladder is just underneath the liver, and the pancreas is just under the stomach. So the functions of the digestive system are all related to food and water. So you have ingesting and digesting food, then absorbing the nutrients from that food, and excreting whatever was not absorbed as waste. Then you also have fluid homeostasis, so that means how much water is absorbed from the gut versus how much water is put out into the gut. And also electrolyte or ion homeostasis, so as water is crossing the gut wall, different ions will be as well, such as sodium. And just as a note, ingesting food means bringing it into the body by the mouth specifically. So anything that enters the body by the mouth is said to have been ingested. And then digesting the food means breaking it down into smaller pieces. And ultimately into individual nutrients that can be absorbed. In order to carry out its functions, the digestive system uses several processes. First, you have ingestion, so that's both a function and a process of the digestive system, so bringing in the food and the water by the mouth. Then you have secretion, that includes exocrine secretions and endocrine secretions. Exocrine secretions for the digestive system would be like saliva or digestive enzymes or stomach acid or mucus. Each of those has a different role, right? So mucus is like protective, digestive enzymes help digest, and saliva helps digest and moisturize. So those are various exocrine secretions of the digestive system. All those secretions are going to end up in different parts of the GI tract. Then you also have endocrine secretion, which would be secretion of hormones. There's actually a lot of different hormones that are secreted by different organs or different parts of the digestive system. We're only going to cover some of them, not even all of them. And they would also have, of course, different functions related to digestion and regulating how the different processes of digestion are occurring and when. Then you have propulsion, which just means moving items like food through the GI tract, from the mouth to the anal canal. For the most part, peristalsis is the type of contraction that is responsible for propelling items through the GI tract. Peristalsis is when you have basically waves of smooth muscle contraction that move along the GI tract and push food ahead of it. So on the next slide, I have some images of that can make it more clear how that's going on. Then digestion. So again, that's also a function of the digestive system as well as a process. There's two types of digestive processes that the digestive system uses. You have mechanical digestion and chemical digestion. In mechanical digestion, you are physically breaking apart the food into smaller pieces, such as when you're chewing the food up. Then you have chemical digestion, where you're actually changing the bonds or breaking bonds within the food molecules to break them into chemically smaller bits. So that would be done mostly by digestive enzymes. Once you have the food broken down into nutrients, the next function is, or the next process is absorption. So also that's a function and a process. In absorption, you're just taking those nutrients, also ions or water, from the GI tract and transporting it into the blood, or for fats into the lymph instead of the blood. Then finally, the last process is defecation, which is how the digestive system excretes its waste, basically pooping. So anything that didn't get absorbed, any nutrients that were not absorbed by the GI tract, will be excreted by defecation as part of the feces. And you also have a lot of bacteria in the gut that are excreted in the feces as well. So defecation can also be considered propulsion, since you are propelling that out of you. So it's kind of hard to categorize. It's like both its own process and a subtype of a different process. And then as a final note, there's also some other waste products that are excreted into the GI tract specifically so that they can be, defecated out. A good example of that would be like some of the hemoglobin or the heme group degradation products, such as stercobilin, that is excreted in the feces and is actually responsible for the brown color. So here's a little more on peristalsis. In this GIF, you can see the waves of peristalsis moving along the digestive tract, and briefly some food moving in there as well. So the food is going to get formed into a ball called a bolus, and that bolus or bolus is what is going to be pushed through the GI tract by peristalsis. In peristalsis, you have smooth muscle lining the walls of the whole GI tract, and you're going to have a couple different layers of it. One of those layers, the fibers are oriented kind of going around the GI tract in a circle, so circling it. When those fibers contract, it's going to pull in, or constrict, basically, constrict the digestive tract. So in peristalsis, you have those muscles contracting to make a constriction that's just in front of the bolus, and that point of constriction, or that muscle contraction, is going to slowly and gradually move down the GI tract. As these muscles release, the next muscles will contract. Then when these muscles release, the next muscles will contract. And the bolus is just going to be pushed slowly through the tract. So now, before we go into more detail on the organs of the digestive system and their function, we'll talk about the peritoneal membrane, which we haven't mentioned since the beginning of ANP1. So the peritoneal membrane is the largest serous membrane in the body. If you remember from ANP1, serous membranes always have two layers, so they're kind of shaped like a beanbag. They're generally going to be wrapped around some type of organ. So it's like if you put an object into a beanbag and then wrap the beanbag around it. The serous membranes are going to secrete a fluid called serous fluid into like the middle space between the two layers. And the basic purpose of it is just to reduce friction so that when organs are moving, they're not rubbing against each other. The peritoneal membrane is covering most of the organs in the abdominal pelvic cavity, just wrapping around them. So it's a really large membrane with its two layers, but then it even has extra folds that are kind of folding around all the organs that are inside of it, and all the little, all the little folds of the small intestine. For the organs in the abdominal pelvic cavity, you can classify them, most of them, as either intraperitoneal or retroperitoneal. the intraperitoneal organs are located somewhere inside those folds of the peritoneal membrane, and the retroperitoneal organs are found behind it or posterior to the peritoneal membrane. Most of the organs in the abdominal pelvic cavity are intraperitoneal, and then some of them are retroperitoneal, or at least part of them is retroperitoneal, and then some of them are also located below or inferior to the peritoneal membrane. So in this diagram here, it's showing you the peritoneal membrane. It's all this stuff in blue, so you can see it has all these folds. And organs like the liver here that are enclosed in those folds are intraperitoneal. So you can see the stomach's also intraperitoneal. Most of the large and small intestine is intraperitoneal. Here's part of the small intestine that is retroperitoneal. Here's part of the large intestine that's retroperitoneal. The pancreas is here, that's also retroperitoneal. The kidneys aren't shown in this diagram, but they're also retroperitoneal. And the rest is mostly intraperitoneal. The bladder sits below or inferior to the peritoneal membrane. Then the layers of the peritoneal membrane have names, and of course every single fold of the membrane has its own name. So for the layers, the outermost layer is called the parietal peritoneum, the innermost layer is the visceral peritoneum, and then the little space between them that's filled with the serous fluid is called the peritoneal cavity. So the visceral peritoneum is always the layer that's up against whatever organs would be wrapped in the peritoneal membrane. The parietal peritoneum would be facing kind of the outside. Then for the folds of the peritoneal membrane, they're called collectively the mesenteries. Most of the mesenteries are around the small intestine, so all these little folds are the mesenteries. Also these are the mesenteries. And you have a lot of places where those folds kind of end up coming together into a ligament that attaches to some more stable structure in the body. And then that ends up kind of being used for structural support to support the organs that are wrapped up in these folds. And those folds will also carry blood vessels or nerves to the different organs that are in them. Out of the mesenteries, each one has its own name. We're only going to learn a few of the names. So first you have the mesocolon, which is the mesentery that wraps around the large intestine. Then you have the greater omentum, which is the mesentery where it's kind of double folded between the stomach and the small intestine. So the greater omentum would actually be right here. It starts at the inferior part of the stomach and then travels further inferiorly to the pelvis. And you have kind of one finger of it here, that's the inner fold of the greater omentum, and then wrapping around that here is the outer fold, or the second fold of the greater omentum. Then you have the lesser omentum finally, which is located right here between the liver and the stomach. And the mesocolon is actually not shown on that diagram. So here you can see the mesenteries again. This view is kind of, I guess, unmanipulated, except that the liver is being pulled up. So in the front, you can see the greater omentum, and you can see it's pretty fatty for protection for the abdominal organs. And then the liver has been lifted up to reveal the lesser omentum stretching between the stomach and the liver. Then in this view, the greater omentum has been lifted up and is exposing the rest of the mesenteries behind it. So all of these long kind of convoluted folds of the small intestine would have a mesentery attached to their other side that's kind of pulling back up into here for structural support. And then this is the large intestine. Most of the large intestine is accounted for by the colon. So this here would be the descending colon. This is the transverse colon. And behind the small intestine here, you would have the ascending colon. Actually, you can see it on this side, the ascending colon right there. So ascending, transverse, descending. So in here is actually the mesocolon, the mesentery that is supplying the colon and giving it support. So not only are these mesenteries kind of supporting the intestines, but you can see the blood vessels that are traveling in the mesenteries through to the intestines. And there would also be nerves traveling adjacent to those blood vessels as well. So now we'll go through the GI tract from the mouth to the large intestine. And as we go, we'll also talk about some of the accessory organs that come up, well, specifically related to the oral cavity. So we'll talk about the anatomy of each organ in turn and its function. First, we'll go over the walls of the organs of the GI tract because they all have really similar layers in their walls. So they all basically have the same scheme, and then there may be some differences or some modifications to the basic scheme for different organs, but the basic format would be there. So the space inside the GI tract is always called the lumen, which is the same as the space inside the blood vessels, also called the lumen. Generally, the space inside of a hollow organ could be called the lumen. Then the innermost layer is the mucosa, which has 3 sublayers within it. First, you have the epithelium, so that's the actual innermost sublayer. Then you have the lamina propria, then the muscularis mucosae. So for the epithelium, if you're in the upper GI tract from the mouth down through the esophagus, the type of epithelium will be non-keratinized stratified squamous. So that's a really strong type of epithelium, for being epithelium, and it allows the upper parts of the GI tract to be better protected from, sharp things that might be in the food, protection from the food. Then when you get to the stomach, then down through the rest of the GI tract, so through the large intestine, the epithelium will change to simple columnar, and that's because you need a thinner layer of epithelium to have proper absorption. So just a single layer of columnar cells lets you get good absorption. And you're also going to have a lot of goblet cells in the epithelium of the mucosa from the stomach to the large intestine. Those goblet cells, you may remember from ANP1, are actually glands. They're single-celled glands, and what they do is secrete mucus as an exocrine secretion. So the walls of the GI tract would be lined on the inside with mucus for most of its, path. Which is to protect it from not only from the food, but also from digestive enzymes, and in fact, particularly from digestive enzymes and stomach acid. So that's the epithelium, the innermost layer. Then you have the lamina propria outside of that, which is composed of loose connective tissue, which makes sense because next to epithelium, you always have loose connective tissue. So the lamina propria is going to be carrying in the blood vessels that are supplying the epithelium with blood. Also, when you have nutrients that are absorbed across the epithelium in the lower GI tract, they're going to be entering into the blood in those blood vessels in the lamina propria. You also have some glands in the lamina propria and MALT, or mucosa-associated lymphatic tissue. So any microbes that came in through the GI tract, if they get kind of into the walls, so if they start penetrating the walls, they may well run into some MALT, and then the immune system would have the opportunity to respond to those bacteria. Then the last sublayer of the mucosa is the muscularis mucosae, which is made of smooth muscle, and you have 2 layers of it. The innermost layer has the muscle fibers oriented circularly, so they're going around the GI tract in a circle, and then the outermost layer has them oriented lengthwise or longitudinally, going along the length of the GI tract. So this diagram shows you the basic composition of the GI tract walls. So far we've talked about the innermost layer, the mucosa. The lumen is the space inside the GI tract, and then you can see it's lined with this red stuff, which is the epithelium of the mucosa. Then in gray, you have the lamina propria of the mucosa, and you can see some glands inside of it, in the green there and like orange. Then outside of that, you have the muscularis mucosae, 2 layers of it, and the outermost layer has fibers oriented longitudinally. This image here is a microscope image of the tissue in the esophagus and stomach, like right at the junction between the esophagus and the stomach. So you can see the epithelium of the mucosa actually transitioning from non-keratinized stratified squamous in the esophagus to the much thinner. Sorry if you heard that, that was my cat. So the epithelium here is transitioning to the much thinner, simple columnar epithelium in the stomach, which allows good absorption. And you can see the goblet cells, these cells in the epithelium that are kind of filled with white. They're actually just goblet cells that are filled up with mucus that they're going to excrete. The next layer in the GI tract wall is the submucosa, which is dense irregular connective tissue and also carries blood vessels, glands, and nerves similar to the lamina propria of the mucosa. Then outside of the submucosa, you have the muscularis externa, which is smooth muscle. And similar to the muscularis mucosae of the mucosa, the muscularis externa also has 2 layers of smooth muscle. The innermost one has the fibers oriented perpendicular to the GI tract, so they run around it in a circle. The outermost layer has longitudinal fibers that run along the length of the tract. And you have nerves coming into the muscularis externa to control those smooth muscle cells. Then the outermost layer of the GI tract wall is called the adventitia or the serosa, depending on where in the GI tract you are. For most of the GI tract, it would be called the serosa. So for all the intraperitoneal organs, they are going to be lined on their outermost surface with serosa, which is actually just the visceral peritoneum of the peritoneal membrane. So that would be composed of loose connective tissue with simple squamous cells lining the innermost surface of the visceral peritoneum, which would actually be facing out in this case if it's wrapped around an intraperitoneal organ. Then for the retroperitoneal organs, instead of having the serosa, they have the adventitia, which is just dense irregular connective tissue, similar to the outermost layer in the wall of a blood vessel. So in this diagram here, you have the rest of those layers, or all those layers. So the mucosa is the innermost. Then you have the submucosa, which is dense irregular connective tissue, and you can see it has glands and blood vessels and nerves. Then you have the muscularis externa, 2 layers of it. The innermost one has circular fibers, and the outermost one has the fibers running lengthwise. Then you have, in this case, the serosa wrapped around the outermost surface, I guess, of the GI tract wall. So this is just visceral peritoneum, and you can see it's carrying in blood vessels, a lymphatic vessel, and nerves. If this was a retroperitoneal organ, it would have the adventitia instead of the visceral peritoneum or serosa. And the last thing we'll talk about related to the GI tract wall is the enteric nervous system, or the ENS, which is all of the neurons and their axons that are located in the GI tract wall. So you actually have a lot of neurons with their cell bodies in the walls of the GI tract. Most of those cell bodies are in the submucosa or the muscularis externa layers of the GI tract wall. Their axons are traveling to sensory cells in the gut, sensory receptors, also to the smooth muscle of the gut, or blood vessels going through the gut wall, or to the secretory cells of the gut, so the cells producing some type of secretion, either endocrine or exocrine. Then those cells are also connected to the nervous system that's coming into the gut. or I should say the parts of the peripheral nervous system that are sending axons into the gut. So that would be the sympathetic nervous system, the parasympathetic nervous system, and the visceral sensory division of the peripheral nervous system. So for instance, if you have stretch in your gut because it's full of food, that is going to be picked up by stretch receptors in the wall of the GI tract. Those receptors are going to be part of the enteric nervous system, or will deliver their signal to the enteric nervous system. The enteric nervous system is going to carry that signal to a visceral sensory neuron in the peripheral nervous system, and then that neuron will send it into the brain. If the brain is going to control the functioning of the gut, for instance, by stimulating peristalsis to help you move your food along, it would do that using the PNS, the parasympathetic nervous system. So you would have parasympathetic signals going into the GI tract wall. They're actually going to contact cells in the enteric nervous system, which will carry the message the rest of the way to the smooth muscle of the gut. The same thing for the sympathetic nervous system. Any messages, any commands to the gut that are carried by the sympathetic nervous system are generally going to contact cells of the enteric nervous system, and then those neurons will relay it the rest of the way to the final effector cell, either a muscle or a secretory cell, endocrine or exocrine. So the motor portions of the enteric nervous system are able to control how blood is flowing through the walls of the GI tract, how the different glands and secretory cells in the GI tract are producing their secretions, how the smooth muscle in the GI tract is contracting, for instance, for peristalsis or for some of the other movements that the GI tract can do, different parts of it. And finally, that can control how nutrients are being absorbed across the walls of the GI tract. In general, the parasympathetic nervous system, or PNS, would be increasing secretion from glands and contraction of smooth muscle and absorption of nutrients and blood flow, whereas the SNS, or sympathetic nervous system, would be decreasing all of those things in order to inhibit the activity of the digestive system. But either way, those messages would be carried through the enteric nervous system. Then the sensory part of the enteric nervous system is able to sense the stretch in the walls of the GI tract and different chemicals that are present in the lumen of the GI tract. Prominently pH, or free protons. It's important for the pH of the different parts of the GI tract to be well regulated. It needs to stay within certain ranges in different parts of the tract. So you need to be able to sense what it is in order to control it. So for instance, if the pH is getting too low in a certain part of the GI tract, you might have that information picked up by a chemoreceptor. It would feed into the enteric nervous system, and the enteric nervous system might carry it to might respond reflexively by decreasing secretion from the glands in the GI tract that release stomach acid. Overall, the enteric nervous system is really important for coordinating the activity of different parts of the GI tract. In order for food to be digested efficiently, you have to have different organs all working together to digest that food. Each organ needs to be doing a different thing at a specific time in order to efficiently digest the food. So the ENS is making sure that different organs in the GI tract can coordinate with each other and work together effectively. In the diagram here, it's showing kind of a summary of the enteric nervous system and the things that it's connected to. So actually on the left side of the diagram, these would be the parts of the peripheral nervous system that are connected to the enteric nervous system. Then on the right side, you have a representation of the GI tract walls and the enteric nervous system. So on the rightmost side, you have the mucosa with different secretory cells in it. Some of them are endocrine cells, so they make a hormone. Some of them are exocrine, so they would make something else, maybe stomach acid, maybe digestive enzymes, maybe mucus, different things. You also have some sensory receptors in the mucosa. These mechanoreceptors would be the ones sensing stretch, and the chemoreceptors here might be sensing pH or some other chemical in the lumen of the GI tract. Then you have the thin muscularis mucosae also in the mucosa. So this part of the mucosa would be the lamina propria and or the epithelium. These secretory cells would be in the epithelium, actually. Then after the mucosa, you have the submucosa. So you have a lot of cell bodies of the enteric nervous system located in the submucosa. All these kind of black balls are cell bodies of neurons in the submucosa that belong to the enteric nervous system. Then after the submucosa, you have the muscularis externa, which is represented here with its two muscle layers, the circular one and the longitudinal one. And you have more cell bodies of the enteric nervous system located there in black and green. So all these neurons belong to the enteric nervous system. You can see their axons are contacting these secretory cells in the mucosa, they're contacting blood vessels, they're contacting smooth muscle in the muscularis externa and in the muscularis mucosae. They're also contacting neurons of the peripheral nervous system that are coming in to the gut wall. So first you have sensory neurons. These would be visceral sensory neurons. So information picked up by these stretch receptors or the chemoreceptors would be relayed to this visceral sensory neuron and be sent out to the brain there. Then you have parasympathetic neurons, some of which belong to the vagus nerve, which would be coming in, sending their axons into the GI tract wall and actually contacting neurons of the enteric nervous system. So any commands that are sent by the parasympathetic nervous system will go to the enteric nervous system, and then the enteric nervous system will relay the command to its final destination, whether that be smooth muscle or some type of gland. And the same for the sympathetic nervous system. Here you have in red the preganglionic sympathetic cells. In blue you have the postganglionic sympathetic cells. So the axons of the postganglionic cells are entering the GI tract wall. Many of them are contacting cells of the enteric nervous system. And then you do actually have a few that would go directly to a target as well, either a gland or smooth muscle. Now for the actual GI tract. The first organ of the GI tract that food would encounter is the oral cavity, or the mouth. In the front, the oral cavity is bounded by the lips. On the sides, it's bounded by the cheeks. On the bottom, bounded by the tongue. On the top, bounded by the palate. And in the back, it just kind of merges into the throat. So the palate has two different parts of it. In the front, or anteriorly, you have the hard palate, and in the back, you have the soft palate. The hard palate is stretched over bone, which is what makes it hard, and it has kind of a corrugated or rough surface, which helps with mechanical digestion. So the tongue can actually push food up against the hard palate, and it will be kind of ground down against the hard palate. Then behind that, or posterior to the hard palate, you have the soft palate, which is tissue stretched over muscle instead of bone, so it's soft. Then coming off of the soft palate, you have the uvula, which is a little flap of tissue that's just designed to cover up the passage up to the nasal cavity when you swallow, so that food will not go up into your nose as you're swallowing it. It'll go down into the pharynx and esophagus. You also have three important accessory organs of the GI tract in and around the oral cavity, which would be the tongue, the salivary glands, and the teeth. And we'll go over each of those in more detail. The teeth, you have three different types of teeth that are each designed for different things, different aspects of mechanical digestion. So in front, you have the incisors that are for cutting food. On either side of the incisors, you have canines that are the sharp ones, so they allow you to kind of rip into food, into tough food. And then in the back you have the molars that have a flat but rough surface, so they're good for grinding. So in the mouth you would have the teeth doing mechanical digestion, of course. You would also have the tongue participating in mechanical digestion by rubbing the food up against the hard palate. So the tongue and the hard palate together would coordinate for mechanical digestion as well. So a little more detail on the tongue. The functions of the tongue are taste, ingestion, and mechanical digestion. So it allows you to have a sense of taste. Most of your taste buds are on the tongue, actually not all of them. It helps you ingest your food and get it further into the GI tract. And it can also push food against the hard palate for mechanical digestion. In the front and at the bottom of the tongue, you have a ligament called the lingual frenulum, which is what is keeping the tongue attached to the bottom of your mouth so you don't swallow it. It's actually impossible to swallow the tongue because of that ligament, as long as it's intact. And you can actually see that if you lift your tongue up, the lingual frenulum will be kind of that line that's sticking out along the bottom of the tongue. You have two types of muscles in the tongue. It's basically mostly composed of muscle. You have the extrinsic muscles and the intrinsic. The extrinsic muscles move the tongue around, and the intrinsic muscles will change the shape of the tongue. The main function of the extrinsic muscles is to help you shape your food for better ingestion, basically making it into a bolus, into a ball called a bolus that then is easy to swallow. Then the intrinsic muscles that change the shape of the tongue allow you to push the food up against the hard palate, which kind of helps break it up for mechanical digestion. The upper surface of the tongue is covered in little bumps called papillae. So those are not actually taste buds, those are papillae. Some of those papillae have taste buds on their sides, but actually most of them do not. You have three different types of papillae as an adult. Most of your papillae are filiform papillae, so those would be all the littlest ones that you have commonly kind of in the front. Those actually don't taste at all, but they do have receptors, like touch receptors for somatosensation. So that's where your sense of touch on your tongue is coming from. Also, if you eat spicy food, that would be picked up. As a type of somatosensation. So the epithelium that you have on the filiform papillae is keratinized stratified squamous, which is actually the same as the epithelium on your skin. It's the hardest, strongest type of epithelium. Because it has that keratin in it, that makes it really hard, and that actually allows the tongue to help further with mechanical digestion. When you're kind of rubbing food against the hard palate, you're also rubbing it against the rough surface of the tongue, which helps break it up. So of course cat tongues are really rough. They have even more keratin in them than our tongues do, but ours still do have some keratin. Then the somewhat bigger taste buds are called fungiform taste buds, and the biggest ones of all are the valate or the circumvallate the somewhat bigger papillae are the fungiform papillae, and then the biggest ones of all are the valate or circumvallate papillae. In this diagram of tongue papillae, you can see the filiform are all of these guys that are kind of streaky almost on their surface. Then the bigger ball-like ones are the fungiform papillae, and the very biggest ones of all, the huge ones, are the valate or the circumvallate papillae. So your taste buds are all on the circumvallate or on the fungiform, but most of them are on the circumvallate, and they're on the sides. So all these kind of little dots along the sides of this papillus are the taste buds. Here you can see another view of them. This whole thing would be a circumvallate papilla, and here you have the taste buds. Then if you were to kind of zoom in even further on a taste bud, this is an individual taste bud. This is like the diagram form that lets you see better. So these highlighted cells specifically are for the taste bud. Some of them are actually sensory cells that would be responding to the chemical composition of the food and sending those messages to nerves that are attached to each taste bud. And then next we'll go into a little more detail on the salivary glands. Of course, their job is to produce saliva. Saliva has a couple different functions. It functions in chemical digestion, and it also retains moisture in the oral cavity. The composition of the saliva differs depending on which salivary glands it was produced from. There's three different types of salivary glands that you have. In general, saliva will have some amount of water, well, it's based on water, and then it'll have some amount of mucus and different enzymes. Mostly it's the parasympathetic nervous system that controls your saliva secretion. So in general, to secrete saliva, you have to have the parasympathetic nervous system send the message to those salivary glands to start producing saliva. Within the salivary glands, you have cells called acinar cells that are actually making the saliva, and there's two different types of acinar cells, which is actually determining the components of the saliva. So first you have serous cells, and then you have mucus cells. The serous cells produce saliva that has a lot of enzymes in it for digestion. The mucus cells produce saliva that have a lot of mucus. So the serous cells will only produce saliva when you're going to be eating, and that saliva has enzymes that help start digesting your food when it's still in your mouth. Then the mucus cells are producing saliva constantly throughout the day. It's a very kind of mucus heavy saliva, so it just kind of coats the surfaces in the oral cavity and just helps retain moisture in there. The three types of salivary glands that you have are the parotid glands, the submandibular glands, and the sublingual glands. You have two of each of those glands, one on each side of the face. The parotid glands are the biggest ones. They're found in the cheeks. They only contain the serous acinar cells, so they can only make saliva that has the enzymes and is produced during eating. Then you have the submandibular glands that are mostly the serous acinar cells, but also some mucus cells. So when this gland is activated, it's going to be producing saliva that has a lot of digestive enzymes, but also some mucus. Then finally, you have the sublingual gland that is mostly the mucus acinar cells. So its saliva is going to be mostly, a lot of mucus with just a little bit of the digestive enzymes. And then this is just a diagram of a zoomed in view of a salivary gland, or part of a salivary gland, I should say, with the acinar cells. These are the serous ones, and then in the paler blue, these are the mucus ones. Then the rest of these cells are just forming the duct of the gland that the saliva would drain into. So we already talked about a couple of the functions of saliva, chemical digestion and moisture retention, but it actually does more things as well. It does help with mechanical digestion just because it's hard to form food into a bolus if you don't have any moisture content to the food. So saliva just kind of moistens things up and helps you form the bolus, which allows you to swallow. And then you also have some immune functions of the saliva because it contains antibodies. So the components of the components of the saliva are mostly water. Like almost all of it is actually just water. Then after that, it has mucus. Specifically, the chemical compound is called mucin, which is for moisture retention. Then it has enzymes. That includes salivary amylase, which is a digestive enzyme that breaks down carbohydrates. Also lysozyme, which is actually not for digestion. It also has immune functions. Lysozyme actually degrades bacterial membranes. So that's an immune function. You also have other enzymes that are in the saliva. Those are just kind of the main ones. Then you also have IgA antibodies, which was the type of antibody that is produced into exocrine secretions. So that's also for immune functions. You also have bicarbonate, or HCO3-. That's actually just in there to neutralize any stomach acid that might happen to come up from the stomach. And a lot more, actually. This diagram is showing all the things that are in saliva. So you can see it's really a lot. Most of it is water, almost all of it is water. But then apart from that, you have a whole bunch of enzymes. The amylase here, that's your salivary amylase. Here's lysozyme. Here's your mucin. So these are other organics, proteins, proteins and such, but not enzymes. Here's your IgA antibodies. Then you also have ions and gases, and here's your bicarbonate. And finally, we'll talk more about the teeth, which of course function in mechanical digestion to help you chew or allow you to chew and break up your food. Teeth are divided into two parts, the crown and the roots. The crown is the part that you can see extending above the gum. The roots are the part that you cannot see because it's embedded either in the gum or in the bone of your jaw or your maxilla. Most of the tooth that is in the crown is enamel. So enamel is like the hard, thick layer on the outermost surface of the crown of the tooth that protects it from the mouth, from bacteria and stuff in the mouth. Enamel is extremely hard. Its structure is very similar to bone, but it actually has more minerals in its extracellular matrix than bone does, which means that it's even harder than bone. So it's actually the hardest substance in the human body. And it can't be repaired, which means that once it is damaged, like for instance by bacteria that are growing on it and secreting acidic waste products onto it, once it's damaged, it cannot be fixed by the body. The body cannot make more enamel. So in order to repair that, you would have to, fill it artificially with a metal or something. Then in the gum and the bone, you have the roots. You have two kind of layers of tooth tissue in the roots. The outermost layer is cementum, which is also very hard, basically as hard as bone. And then within the cementum you have dentin, which is also very hard. It's actually harder than bone. It has more minerals than bone, but it doesn't have quite as many minerals as enamel does, so it's a bit less hard than enamel. So in order from hardest to least hard, it would go enamel, dentin, then cementum. So in this diagram here, you can see the crown on the top with the enamel in it. And then you can see in the roots, the cementum is a really thin layer actually, extending along the edges of the roots. Then beneath that in blue, you have the dentin. So that fills up the rest of the roots, and it actually does extend a little bit up into the crown. Then the innermost part of the tooth is called the pulp cavity. So all this bit in red here is the pulp cavity. Most of it is in the roots, and then a tiny bit extends into the crown. Inside this cavity, you have pulp, which is loose connective tissue, and you have a lot of blood vessels and nerves, as you can see in this diagram. Where the pulp cavity goes down into the roots of the tooth, we call that the root canal. So in a root canal, they would be kind of getting down to this layer and cleaning everything out. The last part of the tooth's anatomy that we'll talk about is the periodontal ligaments, which are these little ligaments that run from the cementum to the bone, either of the jawbone or the maxilla. So either the mandible or the maxilla. These ligaments are anchoring the tooth in place, and that is what you have to break if you're going to extract a tooth. Next, we come to the pharynx, which is basically used to swallow your food. It's the throat. So if we were to get technical, we would say that it functions in propulsion, propelling the food from the oral cavity down into the esophagus by swallowing it. The pharynx can be divided into 3 sections, and actually only one of them is technically considered to belong to the GI tract. So you have from top to bottom the nasopharynx, the oropharynx, and the laryngopharynx. Only the oropharynx is technically part of the GI tract. In this diagram here, you can see the pharynx. On the top here, you have the nasopharynx that opens onto the nasal cavity. In the middle is the oropharynx that opens onto the oral cavity. And then at the bottom of the pharynx, you have the laryngopharynx, which opens onto the larynx, or the airway. So only the oropharynx here is technically part of the GI tract. Inside the oropharynx, you have tonsils, the palatine tonsil here, and the lingual tonsil down by the tongue. Those contain lymphatic tissue and function, have immune functions. So if any pathogens are coming into your mouth and the food that you're eating and they get swallowed, they have the opportunity to get stuck up on a tonsil and activate the immune cells there. Finally, in the pharynx, you have 3 sets of muscles that are used to swallow. And we're not going to worry about the details of how swallowing happens, but these muscles have to contract in a specific order in order for you to swallow properly. First, you have the upper pharyngeal constrictor muscles, then the middle pharyngeal constrictor muscles, and then the lower pharyngeal constrictor muscles, also called the superior, middle, and inferior pharyngeal constrictor muscles. After the pharynx, you come to the esophagus, whose basic job is to pass the food bolus on through it into the stomach, so it functions in propulsion as well. But instead of doing swallowing, it uses peristalsis to move or push the food bolus along the tube of the esophagus into the stomach. The walls of the esophagus contain the esophageal glands, which are basically goblet cells that make mucus. And that mucus just kind of coats the bolus, makes it slimy, makes the walls of the esophagus slimy, helps everything slide on through. The muscularis externa of the esophagus is kind of unique, or it has some unique features in it. At the top, you have part of that muscle that is thickened and forms the upper esophageal sphincter. In this diagram here, you can see that right here, the upper esophageal sphincter, just below the laryngopharynx. So the smooth muscle here can contract to prevent a food bolus from entering the esophagus. So it needs to be relaxed in order to let food into the esophagus in the first place. Then the muscle in the muscularis externa in the top third of the esophagus is actually skeletal muscle, not smooth muscle, which means that the way that the nervous system controls it is different. Then the middle third has kind of a mix between skeletal muscle and smooth muscle, and by the time you get to the bottom third, it's all smooth muscle in the muscularis externa. So in order to pull off kind of peristalsis and move a food bolus from the top of the esophagus to the bottom, you actually have to have multiple branches of the nervous system, both the voluntary motor branches and the visceral motor branches, coordinated to make sure that peristalsis delivers food from the top to the bottom of the esophagus. Then at the very bottom of the esophagus, again, you have thickening in the muscle of the muscularis externa, which is the lower esophageal sphincter, but it's also called the gastroesophageal sphincter because that's the boundary between the esophagus and the stomach. So the muscle in this sphincter needs to relax in order to let the food bolus actually enter the stomach. Once the food bolus has entered the stomach and the stomach is full and is going to be doing its digestion, this sphincter will actually constrict, and that prevents food from rising up from the stomach into the esophagus, and of course prevents stomach acid from rising up as well. So actually this sphincter should in general be closed to prevent food or stomach acid from upchunking into the esophagus and potentially damaging it. Then in this diagram here, or not diagram, sorry, in this microscope picture, in this photomicrograph, that's the fancy name for that type of thing, you can see the layers of the GI tract wall in the esophagus. So first you have the mucosa, this is the epithelium, then beneath that, really thin, you have the lamina propria, then this is actually some muscle that's in the muscularis mucosae. Then you have the submucosa, which is dense irregular connective tissue, and then the muscularis externa. This section is actually from the middle of the esophagus, so some of these cells here in the muscularis externa are skeletal muscle cells, and some of them are smooth muscle cells. The ones that are kind of thick and really vibrant in their color are skeletal muscle cells. Then the smooth ones are kind of really thin. They're really thin streaks, and mostly they're not that deep in color. They're kind of pale. Next, you come to the stomach, which functions in digestion, propulsion, and secretion. So it's propelling food through it into the small intestine. As the food goes through, it is digested by the stomach, both chemically and mechanically, actually mostly mechanically. And you have a lot of secretion going on in the stomach, mostly, of course, stomach acid. So for the anatomy of the stomach, it's, of course, a curved organ. The top of it is called the lesser curvature, and the bottom is the greater curvature. So in this diagram here, the lesser curvature is here on top, the greater curvature is on the bottom. If you look at the interior surface of the stomach, it has ridges on it called rugae, which you can see here in this actual image of a dissected stomach. So those are both basically designed to allow the stomach to expand with food as it fills. Then the stomach is divided into 5 regions. First, you have the cardia, which is directly next to the esophagus. So when a food bolus enters the stomach from the esophagus, it's coming into the cardia. Then you have the fundus, which is the top of the stomach, but not including directly next to the esophagus. Then you have the body of the stomach. Then the pyloric antrum is kind of the bottom of the stomach. And the far end of the stomach where food would exit is the pylorus. So at the pylorus, you have the pyloric sphincter, which controls the movement of food out of the stomach and into the small intestine. The walls of the stomach have several modifications to the basic scheme of GI tract walls. First, the mucosa is unique because it contains structures called gastric pits, which are basically invaginations, or pits in the wall of the mucosa. At the bottom of each pit, you have a gastric gland. Those gastric glands are secreting hormones, gastric juice, which is stomach acid basically, also mucus. The mucus they secrete is very thick because it needs to protect the walls of the stomach from the stomach acid. And the gastric juice is very acidic. The acidity of it will denature any proteins in the food, straightening them out, right, taking away their complex 3D shape, and that helps you with chemical digestion. So if you have a protein that is not denatured, that has kind of its original shape, it's usually kind of balled up to form, some kind of ball-like or clump-like structure. And then if you have enzymes that degrade protein, it's easy for them to access kind of the surface of it, but it's hard for those enzymes to penetrate into the middle of the protein and break down the bonds in the middle. So when you denature the protein and unfold it and straighten it out, then it becomes very easy for digestive enzymes to access the whole part, the whole protein, and degrade it everywhere along its length. So denaturing proteins makes it much easier to digest them later on. Then the other role of the gastric juice is just to destroy pathogens. That's actually its main function. It has a very low pH, and any pathogens that you ate will very likely be destroyed by the extremely low pH of the stomach acid. Let's see, in the diagram here, you can see the gastric pits. So this is all the mucosa. At the bottom, you have the muscularis mucosae. Then you have the lamina propria and the epithelium of the stomach, or of the mucosa. So the lamina propria and the epithelium are kind of arranged to form these little gastric pits, and some of the epithelial cells in those pits are actually forming the gastric glands and secreting all those things. Well, the gastric juice and the hormones. The muscularis externa is also unusual in the stomach because it has an extra layer. So always the muscularis externa will have one layer with fibers running circularly around the GI tract and another layer with fibers running longitudinally along the length of the GI tract. But in the stomach, you also have a third layer that has fibers running diagonally across the stomach, and that allows the stomach to do a churning contraction that mechanically digests the food and basically converts all those food boluses that enter the stomach along with the gastric juice into this kind of fluid called chyme. So the food is essentially getting liquefied inside of the stomach by this churning that the stomach can do because of that extra layer of smooth muscle that it has. And this diagram is showing the process of churning where, the bottom of the stomach will constrict and it kind of makes the fluid chug up in it, and then the top will constrict and the fluid is all getting kind of mixed, and any food bits that are in there are getting mixed as well. As they're getting mixed, they get broken apart until you have just chyme, this liquefied food material called chyme. It's important to form chyme in the stomach because you cannot have a solid food bolus entering the intestine. The intestine is not designed to propel that type of thing. Also, in the intestine is where you have most of your chemical digestion and absorption occurring, and those processes just can't occur efficiently if the food is still solid. It really needs to be liquid so that all the digestive enzymes can get in there and break everything into small pieces, and then each small piece can be taken across the wall of the small intestine. So the stomach's the main function of the stomach acid is to destroy pathogens. The main digestive function of the stomach is actually just that churning of the food to convert it into chyme, although the stomach also plays a role in chemical digestion by denaturing proteins. And we'll talk more about the gastric glands and stomach acid secretion. So in the gastric glands, you have 4 basic types of cells that are secreting something or other. In the upper parts of the gastric glands, you have the mucous neck cells that are secreting mucus. It's very thick, and it's also unusual because it's acidic. Most mucus is actually alkaline, so it has a high pH, but this mucus has a low pH. And the reason for that is so that you don't accidentally neutralize the stomach acid by having an alkaline mucus. Then down in the lower part of the gastric glands, you have the other cell types, one of which is the parietal cell that's actually secreting the stomach acid, which is hydrochloric acid or HCl. The stomach acid has a few different functions. One of the things it does is activate a digestive enzyme called pepsinogen to pepsin. So pepsinogen is the inactive form of that enzyme, and pepsin is the active form. That enzyme degrades proteins. It's also released by different cells in the gastric gland. So the stomach acid will actually activate that enzyme, and it'll start digesting proteins there in the stomach. Another thing the stomach acid does is denature the proteins so that the pepsin can actually access the insides of the proteins and fully degrade them, or have the opportunity to fully degrade them. And then finally, the stomach acid is just destroying any pathogens that may have come into the stomach with the food. In order to release the hydrochloric acid, the parietal cells do a couple of different things. So they actually secrete the hydrogen ions separate from the chloride ions, and then those combine to form HCl, or hydrochloric acid. So the hydrogen ions are excreted using the hydrogen potassium pump, which just pumps out one hydrogen ion, pumps in one potassium ion, and uses an ATP to do that. So you're pumping both the hydrogen ion and the potassium ion against their concentration gradients to where they're more concentrated, so that requires ATP to operate that pump. And then chloride ions will actually just passively follow the hydrogen ions by facilitated diffusion. So they'll just flow down their concentration gradient out of a channel. So here you have a diagram of the gastric gland, and you can see the mucous neck cells are in the upper parts of the gland, secreting the really thick acidic stomach mucus. And then below that you have the other types of cells in the gastric gland. In green, you have the parietal cells that are secreting stomach acid. And here you can see that process where this in the middle would be your hydrogen potassium pump, pumping one hydrogen ion out of the cell, pumping one potassium ion into the cell, using ATP to do that. And then you have chloride diffusing out of the cell through a channel, through a chloride channel. Next, you have the chief cells, which are actually secreting the pepsinogen enzyme. And again, that's the inactive form of that enzyme. Stomach acid will activate pepsinogen to pepsin, which degrades proteins in the stomach. Then you have the diffuse neuroendocrine cells, or diffuse neuroendocrine system cells, to be proper, or DNES cells. They're also called enteroendocrine cells. So they're cells in the intestines and stomach that secrete hormones. So you have some in the stomach, you also have different ones in the small intestine. There's actually a lot of different types of these DNES cells, and they all secrete different hormones. The ones that we have in gastric glands are secreting primarily gastrin and histamine. Both of those hormones stimulate parietal cells. So that could be the parietal cells in the same gland as the DNES cells, or that those hormones could travel in the blood to a different gland and affect parietal cells there. So basically they're just causing the secretion of stomach acid. And in the diagram, all these purple cells are the chief cells that are secreting pepsinogen, and the kind of blue cells are the DNES cells that are secreting hormones such as gastrin and histamine. And we'll conclude our discussion of stomach acid by talking about how it's secreted. So you have three different phases in which stomach acid is secreted. First is the cephalic phase. Cephalic means like of the head. So in this phase, your brain is becoming aware of the fact that there's food around and you're going to be eating, and that stimulates the parasympathetic nervous system via the vagus nerve. And then the vagus nerve and the PNS will stimulate the gastric glands in the stomach to begin secreting stomach acid and the other components of gastric juice. So that would include if you see food, if you smelled food, if you know you're going to eat food. It also includes if you just saw some food on TV or would like to eat some food. All of those things can cause the secretion of some stomach acid via the cephalic phase or in the cephalic phase. Of course, if there's not actually food, then you won't move on to the other phases and you would only secrete a small amount of stomach acid. So if there is actually food that you're eating, then once it enters the stomach, you're going to go to the gastric phase. The stomach will be stretched by the food in it, and that's going to stimulate the enteric nervous system neurons, the stretch receptors in the GI tract wall. Then when the ENS is activated, that's going to activate the gastric glands, and they'll start secreting stomach acid and gastric juice. Then as protein digestion begins in the stomach, because you've had some gastric juice secreted, you're going to get these little bits of protein that are kind of chewed up or chopped up, but they're not fully degraded into single amino acids. So these little partially digested protein bits are also going to actually stimulate the DNES cells in the gastric glands. Then those DNES cells will release hormones like gastrin and histamine, and then those hormones will also stimulate parietal cells in the gastric glands to release stomach acid. So you're going to have even more gastric juice released as a result of protein digestion in the stomach. Then once the food is leaving the stomach and entering the small intestine, you enter the intestinal phase. So you would still have some food left in the stomach at this point, and just some of it is beginning to enter the small intestine as chyme, as like liquefied chyme. So now you have these partially digested proteins in the small intestine, and the proteins in the food are continuing to be digested chemically in the small intestine. So that partially digested protein is also going to stimulate DNES cells that are in the wall of the small intestine. Then those cells release their hormones, prominently intestinal gastrin, and then that causes parietal cells in the gastric glands of the stomach to be activated and release even more stomach acid. So in each of those three phases, the amount of stomach acid that is secreted is like steadily increasing. But it's only going to increase to a certain point. So if the pH of the stomach goes below 2, that is going to be sensed by chemoreceptors of the enteric nervous system, and it's going to signal certain DNES cells to start releasing the hormone somatostatin, and somatostatin is going to inhibit the release of anything else from gastric glands. So it stops the release of pepsinogen and the release of stomach acid. Also, if the pH of the small intestine drops, you're going to have DNES cells there release the hormones secretin and gastric inhibitory peptide, which both will also inhibit the gastric glands. So you would stop making or stop secreting stomach acid and gastric juice. So it's important to have the release of stomach acid stop once the small intestine pH drops, because the small intestine doesn't have super thick mucus like what the stomach has. It has more like normal mucus coating it. So that's not going to be strong enough to protect the walls of the small intestine from damage from stomach acid. As you have kind of a dose of chyme leave the stomach and go into the small intestine, you're going to have some gastric juice enter the small intestine along with it, and that'll drop the pH of the small intestine, and it could potentially damage the walls of the small intestine if you had too much of it there. So once the pH of the small intestine drops because you had some gastric juice enter it, then you're going to have the DNES cells there releasing secretin and gastric inhibitory peptide, and stomach acid secretion is going to stop in the stomach. That way you don't get even more stomach acid, and you can kind of limit the amount of stomach acid that is entering the small intestine from the stomach. And these diagrams are just kind of going over the phases of stomach acid secretion or gastric juice secretion. So on the top you have the cephalic phase, where your brain is becoming aware of food in some way. Then that activates the PNS and through the vagus nerve. The vagus nerve can directly stimulate parietal cells, and it can also activate the DNES cells in the gastric glands, which would then release gastrin and histamine that then stimulate also the parietal cells, causing stomach acid release. Then you would move on to the gastric phase. As food is entering the stomach, that causes the stomach to stretch and activates mechanoreceptors or stretch receptors in the walls of the stomach that belong to the enteric nervous system. So now you have the ENS activating. That is going to stimulate parietal cells. Also, it's going to stimulate the DNES cells that will then release their hormones, gastrin and histamine, also stimulating the parietal cells. Then as these hormones, as the gastric juice is taking effect in the stomach, So you're not just having stomach acid secreted, you're also having pepsinogen secreted, and the stomach acid is activating the pepsinogen to pepsin, so you're going to have some proteins being digested. That gives you partially digested proteins in the stomach. That's going to also further activate the DNES cells to release more hormones to stimulate the parietal cells to make even more stomach acid. And then you move to the intestinal phase, in which the food is now beginning to enter the small intestine. And so now you have your partially digested proteins in the small intestine that are going to stimulate DNES cells there to release their hormones, in this case intestinal gastrin, that then travels to the gastric glands in the stomach and causes the parietal cells to release even more stomach acid. That process is going to continue until the pH of the small intestine actually starts to drop, or if the pH of the stomach gets too low, but in general it would be that the pH of the small intestine starts to drop. At that point, the DNES cells in the small intestine are going to start releasing gastric inhibitory peptide and secretin, which will then go to the gastric glands in the stomach and prevent the parietal cells from releasing more stomach acid. Next, you have the small intestine, which has a lot of functions. It functions in secretion, digestion, absorption, and propulsion. So it's accepting this liquefied food called chyme from the stomach, and it's going to further digest it, actually breaking down all the food molecules into their building blocks. So for proteins into amino acids, for carbs into individual sugars. So in order to do that, it's going to release a bunch of enzymes into the lumen, to digest the food chemically, which means it needs to secrete stuff as well. So you have secretion, digestion, then it's also going to be absorbing those nutrients once they are digested fully, so absorbing the amino acids and the individual sugars and ions and stuff. And then it functions in propulsion, so moving that chyme through the small intestine as it is digested and absorbed. So not only would nutrients be absorbed in the small intestine, but also water is absorbed as well from your food. You actually have some water released into the small intestine also, because the digestive enzymes actually need water in order to operate properly. And in the small intestine is where you have most of the chemical digestion going on and most of the absorption. So the food that enters the small intestine has been digested mechanically a good bit, and it's been digested chemically at least some in the mouth from saliva and in the stomach from pepsin, but just not that much. It's mostly not that digested, chemically speaking. Which means that you have those food molecules in kind of large chunks. They're large molecules still. They haven't been broken down that much. So in the small intestine, they're going to be broken down completely until they're individual building blocks, and then those building blocks are going to be transported across the epithelium of the small intestine and end up in the blood. The small intestine is divided into 3 regions. First, you have the duodenum, which is, coming right off the stomach. It's also the shortest part of the small intestine. Then you have the jejunum in the middle, and the ileum is the last part, and it's actually the longest part as well. In the duodenum, that's where you have your digestive enzymes are secreted into the small intestine. And you have a structure called the major duodenal papilla, which is shown on this diagram right here. It's a little hard to see because it's pretty small, but there is a little circle right there, which is the major duodenal papilla. So the liver and the pancreas are going to be secreting enzymes and other products that are needed for digestion into the small intestine, and all of those secretions are going to enter there at the major duodenal papilla. You also have special duodenal glands in the submucosa of the duodenum, which are producing mucus that's really unusually alkaline. It's very high in pH for mucus, and the purpose of that is just to neutralize any stomach acid that enters the duodenum from the stomach, which there's always going to be there's always going to be some stomach acid entering the duodenum from the stomach anytime chyme is passing into the duodenum. Once food gets to the jejunum, that's where most of your digestion and absorption is occurring. So now the lumen of the intestine is full of digestive enzymes that entered in the duodenum, and they're getting to work digesting the food, and the nutrients from the food are being absorbed. When you get to the ileum, you still have digestion and absorption going on. It's just slowing down. You've already absorbed most of the stuff and digested most of the stuff, so you're just getting what little left that you can out of the food before it passes to the large intestine. So food would go from the ileum into the large intestine through a structure called the ileocecal valve, which is a sphincter that just allows control over how food is entering the large intestine. It's called the ileocecal valve because it's separating the ileum from the cecum of the large intestine. The cecum is like the first part of the large intestine. The walls of the small intestine have some special structures to increase the surface area of the mucosa for better absorption of nutrients from the chyme. The epithelial cells in the small intestine are called enterocytes, so basically intestinal cells. Some of them make digestive enzymes or digestive hormones or make mucus, and all of them absorb nutrients that have been digested, passing them into the blood. So the three structures that you have in the small intestinal wall to increase surface area for absorption are circular folds, which are the biggest, then villi, and then microvilli, which are the smallest. The circular folds are these circular ridges that run around the small intestine. So you'll have the mucosa and the submucosa of the intestinal wall kind of jutting up to form these ridges that are the circular folds. Then within those you have villi, which are like little fingers of tissue poking up from these circular folds. That involves just the mucosa. The villi are going to have in each little finger an artery going down, an arteriole, and a venule coming back, and also a lacteal, a specialized lymphatic vessel called the lacteal where fats would be absorbed. And then in between the villi you have intestinal crypts, so as the little, valleys or pits between the villi are called intestinal crypts. And in the bottoms of them you have special epithelial cells or special enterocytes that are DNES cells that secrete different hormones. Then the microvilli are actually found on the surfaces of the cells themselves. So every enterocyte on its apical surface, which is the top surface, has the microvilli, which are just tiny little projections of the cell membrane. So all three of those structures are increasing the surface area of the small intestine because the surface area is where you can absorb nutrients into the enterocytes. So the more of that you have, the more nutrients you can absorb within the same kind of distance of small intestine. So the small intestine is really specialized for high absorption. So in this diagram you can see those structures. These are the circular folds going around the small intestine. These are the villi. This would be like a photomicrograph of the villi, and here's a diagram of the villi. So you can see in every villus you have blood vessels coming up into it, and you also have a lacteal coming up into it. Then lining it you have the enterocytes, some of which are secreting mucus, some of which would be secreting enzymes. In these intestinal crypts between the villi you have DNES cells secreting hormones. And then if you were to zoom in on any one of those enterocytes, you would see that the top surface or the apical surface is covered with the microvilli, the little projections. You have two types of movements that the small intestine does. First, of course, you have peristalsis to move the chyme through the small intestine into the large intestine. Then you also have segmentation also called intestinal churning, which is a different type of movement. It doesn't propel the chyme through the smooth intestine. Instead, it just kind of mixes it up, which actually helps mechanically digest it. Even though it's almost completely mechanically digested already, you can still do a little bit more. But more importantly, it mixes the chyme with the digestive enzymes that are secreted by the small intestine and into the small intestine from the liver or the pancreas, making sure that you have really good chemical digestion. So as the food is moving through the small intestine, sometimes you'll have peristalsis, actually moving it through the small intestine, and then sometimes the peristalsis will kind of stop, and instead you'll have segmentation going on, mixing the chyme with the enzymes that help it be digested. Peristalsis actually involves not just the circular smooth muscle contracting, it also involves contractions of the longitudinal muscle layer in the muscularis externa. For segmentation, you only have the circular muscle contracting, not the longitudinal. So here you have peristalsis. You can see the circular smooth muscle contracts, also the longitudinal smooth muscle contracts, and this will ultimately push food through the small intestine, or push chyme through the small intestine. Here you have segmentation. Only the circular smooth muscle will contract for segmentation. And the result of this is that food doesn't move through the small intestine. Instead, it just mixes up with itself. The last structure, or the last organ in the GI tract, is the large intestine, which functions in propulsion, absorption, and defecation. So moving the food through the large intestine and then passing it out the other end would be defecation, and also absorption. So by the time the food gets to the large intestine, most of the nutrients have already been extracted from it. Almost all of them have been extracted. So for the most part, the only thing that the large intestine is absorbing would be water and ions. You actually have a good bit of water added to the chyme in the small intestine because the digestive enzymes that will digest the nutrients only work if there's water present. So you have to have water to power digestion. Then you need to reclaim that water in the large intestine. Almost all of it needs to be reabsorbed. Otherwise, you're going to be losing a lot of water in the feces, and that's just not sustainable. So to conserve water, you need to reabsorb it in the large intestine, and anytime you're reabsorbing water, you're generally going to be absorbing ions as well. There's 3 regions of the large intestine. First, you have the cecum, which is very small actually, right where the small intestine empties into the large intestine. That's where you have the cecum, and it really does not extend appreciably far past that point. So technically, what it would be receiving from the small intestine is chyme, that liquefied food. But actually, as soon as it enters the large intestine in the cecum, we don't call it chyme anymore, we call it feces, because nutrients have already been extracted from it and absorbed by the small intestine. So chyme is nutrient dense, feces is nutrient sparse, since the nutrients have already been extracted. So here you can see the ileocecal valve opening into the cecum. This little worm-like thing coming off the end of the cecum is actually the appendix. And from the cecum you would move into the colon. So most of the large intestine is just the colon. And the colon itself has 4 different sections. First you have the ascending colon, then the transverse colon, then the descending colon, and then the sigmoid colon, where it makes a small jog before bending and becoming the rectum. So the very last part of the large intestine is called the rectum, and that leads to the anus. Within the rectum, technically the very last part of the rectum is the anal canal, which you can see in this zoomed in view here. And then guarding the entrance, or I guess the exit of the anal canal, you have the internal and external anal sphincters. The internal anal sphincter is formed by smooth muscle of the muscularis externa. The external anal sphincter is formed by skeletal muscle of the pelvic floor. So that's under voluntary control because it's skeletal muscle. Whereas the internal sphincter is not voluntarily controlled. That's under involuntary control because it is smooth muscle. You have quite a lot of bacteria in your gut, and actually most of them are living in the large intestine. You do have some in other parts of the GI tract, but by far the biggest portion of them are in the large intestine. There's actually so many bacteria in your large intestine that you're kind of constantly excreting their dead cell bodies in the feces. Actually, about 60% of the mass of feces on average is just bacterial cells. Not even food waste, just those bacteria. So there's quite a lot of them. They're called collectively the gut flora. Flora is generally like referring to plants, whereas fauna is generally referring to animals. In this case, we've chosen to call these bacteria flora, even though they're not plants. But gut flora would be just all the microbes in the large intestine, and most of them are bacteria. You can also call them the intestinal microbiome. In general, it's a good thing that they're there. They're helpful for you. There are some kind of, I guess, side effects to having them there. For one, they cause flatulence. So if you didn't have bacteria living in your gut, you actually wouldn't fart ever. The gas of the flatulence is actually waste products that are created by these bacteria as they're doing their metabolism. So if there's like a particular food that makes you gassy, what that actually means is that the gut flora are able to eat that food, or at least certain species are able to eat that food, and it's real good food for them. So they eat it, they do a lot of metabolizing, they make a lot of waste products, and then you have a lot of flatulence. But in general, it is, a good thing that they're there. So they're all living in a symbiotic relationship with us, and most of them are living in a mutualistic symbiotic relationship, which means that they are helping us and we are helping them. Some of them would also be living in a commensalistic relationship with us, where we are helping them and they're not hurting us or helping us. They just have no effect on us. But most of them would be mutualists, so that we're helping each other. You do have some that could be parasitic sometimes, which would mean that we're helping them and they're hurting us. In general, there should not be parasitic bacteria or pathogenic bacteria in your gut flora at any significant level. Like you might have a couple living in there, but as long as there's just a couple of them, that's not enough to actually meaningfully hurt you. If they kind of proliferate and there gets to be a lot of them, then that could cause some problems for you. Also, sometimes if the bacteria in there that are helpful for you kind of leave the large intestine where they're supposed to be at, then sometimes they could cause an infection. So for instance, if you had like a tear in the wall of your large intestine and some bacteria escaped into your abdominal pelvic cavity, that could potentially cause a serious infection, even if they were species that are helpful for you and not harmful at all, as long as they're living in the large intestine like they're supposed to. There's a lot of things that can affect the gut flora. Of course, your diet affects them. Certain things that you can eat would favor certain bacteria over others, and that would change kind of the makeup of your gut flora. There's also some people that need to kind of avoid certain foods because of their gut flora, and prominently that would be people who are lactose intolerant. They're not able to digest lactose, so all of the lactose in, for instance, milk that they would drink was going to pass through the small intestine unaltered, and all will end up in the large intestine. And then you have bacteria in there that are able to eat to digest the lactose. So they digest it they have kind of a population boom because of this inrush of food, and they make a lot of waste products, and that can cause gastrointestinal symptoms that are associated with lactose intolerance. Also, anytime that you're on antibiotic drugs, that is going to affect your gut flora as well. And it's going to change the balance of species in the gut flora. Any species that are harmed by the antibiotic that you're on are going to be killed, and any species that are not harmed are going to proliferate and can reach kind of a larger population than they otherwise would. And then you could end up with like an imbalanced microbiome in your gut where there's too many of some species and not enough of others. Potentially that can also allow pathogens to prosper if those pathogens are resistant to the antibiotics, whereas some of the harmless or good bacteria are not resistant. So that does happen sometimes. Also, the gut flora is kind of a hotbed for the development of antibiotic resistance. Since you do have these bacteria living in there, anytime you take an antibiotic, they're going to be exposed to it, and not all of them will die. So it's not uncommon for some of those bacteria to gain a mutation that gives them resistance to an antibiotic, and then the next time you take that antibiotic, those bacteria will not be harmed by it. So they're at an advantage because they're not harmed by this antibiotic, and then they can proliferate and become more common. So you can actually establish antibiotic resistant strain in your gut. And then sometimes, depending on the species of bacteria and depending on where in the genome this mutation has occurred that gives them resistance, sometimes it's possible for bacteria to share that gene that gives them resistance with other bacteria, even of different species. So in the gut, you can have antibiotic resistance spreading quite widely between different species of bacteria. As soon as one gets it, it's sometimes possible for others to get it as well. And some of those others might be pathogens that are passing through the gut, which would be bad. One thing you can do to kind of rebalance your gut flora if it's become imbalanced is to eat probiotics and also fermented foods. So I guess eating fermented foods would be kind of the more natural way of rebalancing your gut flora. Essentially anything that is fermented is going to be helpful because all fermented foods, well, I should say most fermented foods are created through using these beneficial bacteria that are also good to have in your gut. So stuff like kefir, kombucha, kimchi, those are all fermented foods that have a lot of beneficial bacteria in them, that, it can be very healthy to eat those things or add them to your diet. In this diagram, it's just kind of showing an example of some beneficial species that you can have in your gut flora. Lactobacillus species, E. coli. There is one strain of E. coli that is quite dangerous, but in general, most E. coli strains are not dangerous. They're actually good for you. Bifidobacterium. Another one that's not on here would be Bacteroides. Those are all very common to find in the gut flora, and they're, it's a good thing when they're there, in general. If the gut flora becomes imbalanced, you might see higher levels of some pathogens like Staphylococcus aureus or Salmonella or Clostridium perfringens. Some of these are also commonly resistant to antibiotics. And just a little bit more about the gut flora. Looking at the symbiotic relationship between them and us, what they're getting out of this relationship is basically a nice place to live. There's always kind of food coming by. They don't have to do anything to get that food. And the environment is just very stable. It's always a nice temperature. It's always a nice pH. They never have to worry about changing environmental conditions. So it's a really nice sheltered place for them to be living. What we get out of the relationship is a variety of different things, and it it does depend on like which species exactly we're talking about. So some species that are in the gut flora actually manufacture vitamins that we're not able to produce ourselves, so we need to get them from our diet or from our microbiome. So the large intestine can absorb those vitamins that bacteria produce for us. Also, there's some things that we might eat that we can't digest that bacteria can ingest for us. A good example would be fiber. You have some species in the gut flora that are able to digest that into simple sugars. If you break fiber down, it just turns into simple sugars, but we don't have the enzymes to do that. So then our large intestine could then absorb those sugars after the fiber has been digested by bacteria in the gut flora. They also help control the levels of pathogens. So if you've eaten something, that had some pathogens in it, ideally they should be killed by the stomach acid. But if there was really quite a lot of pathogens in it, then they might not all have been killed. So some would make it through into the large intestine. And there it might be a nice place for them to live, but if they're going to live and prosper there, they're going to have to compete for resources with all of the bacteria that are already living there that belong to your gut flora. And so that means that usually when pathogens enter the large intestine, they can't really get established. So you might have a few of them in there, but they're not going to proliferate to a large level and form a large population. They just aren't able to compete well enough with your natural gut flora to do that. And then finally, the gut flora strengthen our immune system. It helps the immune system develop as you're kind of an infant and a child. The gut flora help the immune system tell the difference between self and foreign. And actually your immune system will learn the antigens of your gut flora, and it will learn that those are self antigens, even though they're not, they're not the same organism as you, but they are inside of you, and it's good for them to be there, so the immune system will just identify them as belonging to you, so that it won't, I guess, trigger an immune response against them. If the immune system were to activate an immune response against your gut flora, that would be a bad thing for you, because not only would it be killing these bacteria that are good for you, it would also be causing, a lot of inflammation in your gut, which would interfere with digestion and make you very uncomfortable. Another thing that the bacteria in the gut flora do for you immune-wise is just help develop the MALT, the mucosa-associated lymphatic tissue. It doesn't develop properly without the gut flora, and you have a lot of MALT in your gut, in the lamina propria. And it's important for it to be there because you have a lot of different microbes passing through the gut. So having a lot of MALT around that can respond to those microbes is really beneficial for you and can help you respond to potential pathogens more quickly than otherwise. And the last thing we'll talk about with the large intestine is its motility, because it's different from the small intestine. So as far as how the large intestine is moving, you can divide it into two different portions, the proximal large intestine and the distal large intestine. The proximal large intestine includes the ascending and the transverse colon. The distal includes the descending colon and the sigmoid colon. In the proximal large intestine, you have two types of movements that occur. First, you have segmentation, which works just the same as it does in the small intestine. But in the small intestine is for a bit of mechanical digestion and for mixing food with digestive enzymes. In the large intestine, the segmentation just increases absorption. And again, the main thing that's being absorbed there is water and ions. Basically, by mixing things around in the large intestine, it helps you absorb everything that you wanted to absorb. Then in addition to segmentation, you have peristalsis, but it's like a kind of a special type of peristalsis that we call mass peristalsis or mass movement. And that's just because it's not happening all the time. So for the small intestine, it's actually always undergoing peristalsis or segmentation, one or the other. And if you have the sympathetic nervous system activate, then the peristalsis is going to slow down in the small intestine, but it's not actually going to stop ever. In the large intestine, you usually don't have peristalsis occurring. You would just have maybe some segmentation. But then just a few times a day, you're going to have the entire proximal large intestine going through these large waves of peristalsis called the mass peristalsis. And the purpose of that is just to move the material, move the feces further into the large intestine, from the proximal into the distal large intestine. Then in the distal large intestine, you don't have very much movement at all. Most of the time it's completely still and it's just acting as kind of a storage depot for the feces. So you're going to have feces kind of accumulating there as the mass peristalsis moves it into the distal large intestine. Then also every time you have a mass peristalsis movement in the proximal large intestine, you're going to have a defecation reflex in response in the distal large intestine. The defecation reflex is moving the feces out of the distal large intestine, first to the rectum and then through the anal canal and out of the body to be excreted. So this is going to happen every time you had mass peristalsis, but that doesn't mean that you're actually going to defecate every time that it happens, because first, of course, you're just going to be moving the feces into the rectum. And of course, you have your external anal sphincter that you control, so you're not just at the mercy of your defecation reflex. It's important for the body to regulate how the feces is moving through the large intestine to make sure that the correct amount of water is reabsorbed. If you have too much motility in the large intestine, feces is moving through it too quickly, then you're not going to be able to absorb enough water out of it, just because it's not spending enough time in the large intestine to absorb all the water that you need, and you end up with really runny feces or diarrhea. That can also end up dehydrating you. So if you're having diarrhea, that means you need to increase your water intake to account for all that extra water you're losing in the diarrhea. On the other hand, if the feces moves too slow through the large intestine, you're going to absorb too much water, and the feces will become really hard and compacted, and at that point it's difficult to move it through the large intestine and out through the anal canal. So at that point you'll have constipation. So one thing that you can do for constipation is to take pills that help incorporate more water into the feces, just to soften it up, make it easier for it to move. And the diagram is showing the defecation reflex, where once you have feces moving into the rectum, it will stretch the wall of the rectum. Then you have stretch receptors there that are activated, that send their information along the visceral sensory pathways of the peripheral nervous system into the spinal cord, where it's going to synapse onto a motor neuron in the parasympathetic nervous system that's headed out to the rectum, and it's going to cause the smooth muscle in the walls of the rectum to contract, pushing the feces out. At the same time, it's going to cause the internal anal sphincter to relax so that feces can pass through the anal canal. But if you're actually going to defecate, then at the same time you also need to relax the external anal sphincter, which is under voluntary control. So you have to decide that it is that time in order to complete the defecation. So now we'll talk about the three accessory organs that we didn't mention yet, which are the pancreas, the liver, and the gallbladder. So these are all going to be adding components to the small intestine that will help with digestion. So first, the pancreas, which as I kind of mentioned, it functions in secretion. It secretes both hormones, so it has endocrine secretion, and it secretes exocrine substances, mostly digestive enzymes, so it has exocrine secretion as well. The hormones, well, the main hormones that are secreted by the pancreas are insulin and glucagon. So those are not directly related to digestion, but they do have kind of, I guess, ancillary roles, where insulin is reducing your blood sugar by causing cells to take up glucose from the blood, and glucagon is increasing your blood sugar by causing the liver to increase breakdown of glycogen into glucose. So those hormones are released by cells that are found in little clusters called pancreatic islets. Then you also have acinar cells in the pancreas that are producing the exocrine secretions, or the digestive enzymes. And it's those exocrine secretions that are secreted into the small intestine through the major duodenal papilla. So each of those acinar cells is going to be releasing its secretions into these ducts called pancreatic ducts, which will merge together to form a large main pancreatic duct that is actually carrying those secretions all the way to the major duodenal papilla. And the primary way that those secretions are triggered is in response to the hormone cholecystokinin, or CCK, which is released by DNES cells in the small intestine. So you would have these DNES cells in the small intestine releasing CCK that travels in the blood to the pancreas, and it causes these acinar cells to release their exocrine secretions, which then travel in the pancreatic ducts. Those ducts merge into the main pancreatic duct, which empties into the major duodenal papilla. So now those secretions are released into the small intestine, and the digestive enzymes can begin digesting the food, or the chyme. Then you have the liver and the gallbladder, which both function in secreting bile. And the liver also has a lot of other functions that aren't directly related to digestion, which we will briefly go over in a bit. The liver is very large, the gallbladder is very small, and it's kind of tucked underneath the liver. The liver has a layer of connective tissue around the outside of it called the capsule, and it's divided into 4 lobes. On the inferior surface of the liver, where the gallbladder is nestled, you have something called the porta hepatis, which is where all of the vessels that need to get in and out of the liver are actually coming in and out. So you have the blood vessels coming in and out there. That actually includes the hepatic artery and the hepatic portal vein. The hepatic vein does not come out of the porta hepatis. It exits on a different part of the liver and drains into the inferior vena cava. Then you have lymphatic vessels also coming in and out of the porta hepatis. Well, I guess technically out, not in. And you have nerves coming in and out. And finally, you have the common hepatic duct coming out. So the exocrine secretions, or the bile that's produced by the liver, all drains into the common hepatic duct and leaves the liver there at the porta hepatis. Some of that bile is going to end up stored in the gallbladder, and then later it would be released. So in the diagram, you can see a bit of the capsule that covers the liver, and some of the lobes of the liver. On the inferior surface here, you can see, here's another lobe of the liver, more of the capsule, and you can see the porta hepatis right here. So you have the gallbladder right next to the porta hepatis. In the anterior view, you can just see it underneath the liver. In the inferior view, you can see it better. Here in green, you have the common hepatic duct. It's actually just called that before you get to the gallbladder, and after the gallbladder, it has a different name, the common bile duct. Then in red, you have the hepatic artery entering the liver, and in blue, the hepatic portal vein entering the liver. You would also have lymphatic vessels and nerves going in, but they're not shown in this diagram. Then on the posterior surface of the liver is where you have the hepatic vein emerging and joining with the inferior vena cava. If you were to zoom in on the liver, it's made of these microscopic structures called liver lobules, or lobules. So the liver lobule is the functional unit of the liver, which means that every liver lobule is able to do the basic functions of the liver. And then you put a lot of those together and you get a complete liver. They're shaped like little hexagons. In the middle of each hexagon, you have a central vein. The central vein is going to drain into the hepatic vein that then drains to the inferior vena cava. At each corner of the hexagon, you have structures called portal triads. Every portal triad has 3 vessels in it, or ducts. It has a hepatic arteriole, which comes from the hepatic artery. It has a portal venuole that comes from the hepatic portal vein, and it has a bile duct that's going to the hepatic bile duct. Then you have capillaries that are connecting the hepatic arterioles and the portal venules to the central vein in the middle. They're sinusoid capillaries, so those are called hepatic sinusoids. The sinusoid capillary, as you remember, has large holes in the walls of it so that cells can actually get in and out of the capillary pretty easily. And it's just very easy for substances to pass in and out of those capillaries. So you have blood from the hepatic arteriole and from the portal venules mixing together in these hepatic sinusoids and kind of mingling with the liver cells. The liver cells are going to be metabolizing the components of the blood, especially stuff that was absorbed from the gut that would be traveling in the portal venuole, just kind of filtering and processing it, removing any toxins. And ultimately all of that blood is going to drain into the central vein and then go out on the hepatic vein to the inferior vena cava. Then you also have little vessels called bile canaliculi that are running through the liver lobule, where the bile is basically being collected from the liver cells. And the liver cells are called hepatocytes. So the hepatocytes are manufacturing bile and secreting it, and is being collected in these bile canaliculi that drain to the corners of the hexagon into the bile ducts at each corner. And then those bile ducts will merge and drain into the hepatic bile duct, or the common hepatic duct. And then finally, surrounding each liver lobule, you'll have the septa, which is just a covering, a connective tissue covering that comes from the liver capsule. So the liver capsule is going to have like little sheets coming off of it that kind of penetrate into the liver and wrap around every liver lobule. So in this diagram, you can see the structure of a basic liver lobule, where it's shaped like a hexagon. It's composed out of these stacked hepatocytes or liver cells. In the middle of it you have the central vein, and at each corner you have a portal triad. The portal triad has a hepatic arteriole, hepatic portal venuole, and a bile duct. The arteriole and the portal venuole have sinusoid capillaries emerging from them that drain ultimately into the central vein. And then that goes to the hepatic vein and to the inferior vena cava. Then you also have these bile canaliculi in green that are collecting bile from the hepatocytes and draining into the bile ducts at each portal triad, and that is going to drain into the common hepatic duct. Then this is a photomicrograph of the liver, so a section of liver tissue, and you can see the liver lobule that's basically a hexagonal structure, and you can see the portal vein, the central vein in the middle of it. If you zoomed in more, you would be able to see the portal triads. Those vessels are a bit smaller, so they're harder to see in this view. So the function of the liver that's directly related to digestion is producing bile. And the function of the gallbladder is to store that bile and concentrate it. So in the liver lobules, you have the hepatocytes making bile, which drains into the bile canaliculi, and then that drains into the bile ducts in the portal triads, and then those bile ducts all drain into the common hepatic duct. The common hepatic duct is going to take the bile to the gallbladder, where it is stored until it's triggered to be secreted. The function of bile is twofold. First, bile is going to kind of emulsify any lipids that are in the chyme, which allows them to be absorbed. And second, the bile is going to have some wastes in it that it's carrying that are, to be excreted. So any waste that is in the blood that is not going to be excreted by the kidney is probably going to be excreted in the bile. So the liver is going to take those wastes and put them in the bile as the blood is passing through the liver lobules. And then, of course, the waste would be carried with the bile into the small intestine and would remain there and exit in the feces. A big example of a waste that is excreted that way is bilirubin from the degradation of heme. So that is put into bile by the liver. Some of it is actually reclaimed ultimately and is excreted by the kidneys as urobilin, and the rest of it is excreted in the feces as stercobilin, and it gives the feces its brown color, actually. So in addition to including wastes or containing wastes, the bile has compounds called bile salts. The bile salts are derived from cholesterol, so their structure is pretty similar to cholesterol. Cholesterol is a type of lipid that has 4 rings made out of carbon in it. Bile salts also have things added to that cholesterol base that are polar or charged, which makes the bile salts amphiphilic. So they're lipophilic on one end, or hydrophobic on one end. On the other end, they are hydrophilic. So on one end, they interact with lipids. On the other end, they interact with water. Amphiphilic just means that it's a compound that is hydrophobic on one end and hydrophilic on the other end. So you might remember that term from general biology, maybe not, but it comes up when we're talking about the cell membrane because the phospholipids that make up the cell membrane are amphiphilic as well. So they have these long fatty acid tails. Those are lipophilic or hydrophobic, and then the heads have polar or charged groups on them. So those are hydrophilic. And when the phospholipids come together to form the cell membrane, you get all the hydrophobic tails pointing in towards each other in the internal interior of the membrane, and the hydrophilic heads are pointing out on either side to the cytoplasm of the cell or to the extracellular fluid. Soap also works this way. It's amphiphilic, so it has part of it that will interact with water, part of it that will interact with lipids. And that's what allows soap to cut through grease and, help you clean grease off of things. So water cannot interact with grease directly because water is hydrophilic, of course, and grease is hydrophobic. So the soap is going to interact with grease on one end and kind of grab the water on the other end, and that lets you get that grease off. So bile salts are working very similar to how soap works. They interact with lipids in the chyme on one end and interact with water that's in the intestine on the other end, and that allows them to break large kind of globs of lipids up into smaller and smaller bits. We call that emulsification. Emulsification is when you have two things that are not going to mix together, like oil and water, and you add something to make them mix or to emulsify them. So you're going to get all these little fat droplets that are surrounded with bile salts on all sides, and that are emulsified in the water by those bile salts. A common example of an emulsification is just milk. So in this image here, you have kind of that process. So you start with these kind of large globs of fat in the chyme. Since fat does not interact with water, it's hydrophobic, it's not going to kind of get broken up in the water very much. It's going to stay in these large globules where the lipids are all kind of just sticking together there. But when you add these bile salts that are hydrophobic on the yellow end and hydrophilic on the green end, the bile salts are able to kind of get into this fat globule and start breaking it up. They will surround the little particles of fat on all sides with their hydrophobic portion interacting with the fat droplet. Then their hydrophilic portions will interact with the water surrounding the globule. And you can get a lot of really small emulsified fat droplets by adding those bile salts. If they're large, if the fat globs are like too large, then it's impossible to absorb them. You really have to break them up in order to absorb them. So you need the bile salts to do that. And then this diagram is just showing how the gallbladder is kind of related to the different bile ducts. So you have in the liver all your little small bile ducts that kind of merge together and ultimately merge to form the common hepatic duct. So bile is going to be traveling in this common hepatic duct, and then it's going to back up kind of into the gallbladder and be stored there and also concentrated until it's time to release it. And finally, we'll talk a bit about how bile is actually secreted. It can be secreted in response to two kind of triggers. One of them is the hormone CCK that comes from the small intestine, from the D and ES cells of the small intestine. And the other trigger is just having too many bile salts in the gallbladder. So if the gallbladder kind of recognizes that it's full of bile salts, then it will release bile. Also, if you have the hormone CCK circulating to the gallbladder, it will trigger it to release its bile. When the bile is released, it's going to travel along the cystic duct from the gallbladder. The cystic duct then merges with the common hepatic duct to form the common bile duct. So the bile is going to drain into that common bile duct, and that's going to carry it most of the rest of the way to the small intestine. Just before it's about to enter the small intestine, you have the common bile duct merging with the main pancreatic duct from the pancreas to form the hepatopancreatic ampulla, which has a little sphincter around it, the hepatopancreatic sphincter of smooth muscle. So that's the last opportunity to kind of control how bile salts, or how bile and how digestive enzymes from the pancreas are entering the small intestine. So if that sphincter is relaxed, then the bile will, journey the short rest of the way and enter the small intestine at the duodenum through the major duodenal ampulla. Then the bile salts are going to mix with the chyme, emulsify any lipids that are in the chyme. Ultimately, they're going to be reabsorbed by the ileum of the small intestine, which is the last part of the small intestine. They're going to end up draining into the hepatic portal vein and going back to the liver. So your bile salts are mostly going to be recycled. You have a few of them that are excreted in the feces, but most of them you hang on to. And of course, any waste products that were with the bile are going to be excreted in the feces and not reabsorbed for the most part. Then we'll also go over a couple of the major liver functions that are not directly related to digestion. Both of these functions are kind of indirectly related to digestion, but they don't directly impact it. So first you have metabolic functions of the liver, where the liver is kind of getting the first crack at all the nutrients that have just been absorbed by the intestines. So all those nutrients are absorbed into the blood. That blood will all drain into the hepatic portal vein and be carried to the liver lobules. So those nutrients are all distributed amongst the liver lobules, and the hepatocytes kind of get the first chance to use those nutrients. They're going to take a lot of the sugars that have been absorbed from the intestines and use them to make glycogen, which is kind of how the liver is storing sugar. It's kind of a large polysaccharide molecule, and anytime that you need extra sugar, the liver can break down that glycogen and release more sugar for you. Liver would also be using those nutrients to make albumin and a lot of the other plasma proteins for the blood, and a variety of other compounds that the liver makes. Then whatever nutrients the liver didn't use are going to pass through it and ultimately end up in the inferior vena cava to be distributed to the rest of the body. The liver also has a function in detoxifying or excreting things. So substances in the blood that are not going to be excreted from the kidneys could be excreted from the liver by being put into bile. And then for detoxification, some things that you may have just eaten and absorbed from the intestines might not be so good for you. Those things will be detoxified by the liver and then excreted. either by the liver or by the kidneys in urine. So a very wide range of molecules can be detoxified by the liver. In general, the liver is always taking some toxin that is non-polar, lipid-soluble, hydrophobic, and converting it into something that's polar, that is water-soluble, because if it's water-soluble, then it's easy to excrete it in the urine or in the bile. So the liver is not necessarily reducing the toxicity of the toxins that it detoxifies, but it's always making it easier to excrete those substances. Sometimes the liver actually might make something more toxic by detoxifying it. Sometimes, the compound the liver converts something into is actually more toxic than the initial compound that you started with. But we still call that a detoxification because this compound that's more toxic is also going to be more water soluble, which makes it easy to excrete. If you have something that's non-polar in the blood that's a toxin, it's pretty hard to get rid of that because only water soluble things will really go into the urine. So you might be able to put some of those things into the bile because of the bile salts, but apart from that, you can't really get rid of non-polar toxins. So you have to make them polar before you can excrete them. So the liver is doing that for us. And in this diagram, it's kind of showing a general schematic for how the liver detoxifies things. So you have your toxins entering the liver. They would be generally non-polar things that do not dissolve well in water. Then the liver is going to have kind of two phases of detoxification. The first phase is catalyzed by cytochrome P450 enzymes, or the CYP enzymes, which you may have heard of. They're responsible for metabolizing, all these toxins, but also a bunch of different drugs, like, prescription drugs and over-the-counter drugs. They're going to convert these toxins or these drugs into intermediate that is more water soluble than what it started as, but still probably not fully water soluble. And then you have phase 2, in which the liver is going to take some other polar compound and attach it to that intermediate. So that's called conjugation, because you are taking two things and attaching them together. The result of that is going to be something that's quite polar, very water soluble. That is going to be excreted probably by the kidneys in the urine, but maybe also by the liver itself in the bile. And down here you have an example of a compound that is frequently conjugated to intermediates in the liver, which is glucouronic acid. So it has all these OH groups, and it's not a symmetrical molecule, so that makes it polar. So it's actually quite polar. Also, if you put it in water, some of these OHs will lose their H and pick up a negative charge. So when you attach this glucouronic acid to something that's kind of maybe weakly polar or a bit polar, it becomes very polar because this is so polar and it has charges on it in water. And that's going to make that thing very water soluble probably. Then in blue here you have free radicals and reactive oxygen intermediates. These are both just kind of different types of reactive oxygen species. So as your CYP enzymes and your conjugation enzymes are creating these, or I guess detoxifying these toxins and carrying out phase 1 and phase 2, they're going to generate some amount of reactive oxygen species. Reactive oxygen species contain oxygen and they have unpaired electrons, and they're just very reactive, as the name suggests. So they can react with DNA real easily. If that happens, they're going to damage the DNA. They can react with proteins, which would commonly damage those proteins. So if you have a lot of reactive oxygen species around, that's going to cause tissue damage, secondary tissue damage. That means that you need to have a lot of antioxidants in the liver to protect it from those reactive oxygen species. An antioxidant is just something that is going to react with the reactive oxygen species before something else can. And once you've had that reactive oxygen species react, usually it's not toxic anymore after that. It's not reactive anymore after that. So antioxidants will react with the ROS first, and that protects you. So that's one reason to have a lot of antioxidants in your diet. It protects your liver from the ROS, or the reactive oxygen species, that it generates when it's detoxifying things in the blood. I guess a final thing to mention here is the CYP enzymes, the cytochrome P450 enzymes. You have a lot of drugs that will affect those CYP enzymes, and that causes interactions between those drugs and other drugs that are metabolized by the CYP enzymes. So if you have a drug that deactivates a CYP enzyme as like a side effect, and you're also supposed to be taking a different drug that is metabolized by the CYP enzyme, that CYP enzyme isn't going to be as active because of the first drug that you're on, and that means that the second drug you're on is going to last for longer in your system, and it may accumulate to higher concentrations in your blood. So that might be dangerous depending on what the drug is, so that might mean that you need a lower dose of that drug. And this is also the reason why, if you're on certain drugs, you might be told to avoid things like grapefruit juice. Grapefruit juice has compounds that actually inactivate certain CYP enzymes that are responsible for degrading a lot of different drugs. So if you're on the grapefruit juice and you're taking one of those drugs, or if you're drinking grapefruit juice and taking one of those drugs, you might end up with more of the drug in your system than you should have. Finally, for the liver, we'll just go over the hepatic portal system again. So again, a portal vein is a vein that is connecting two capillary beds. So you have the capillary beds in the stomach and the small intestine, also actually the spleen and the pancreas, and large parts of the large intestine. All of those capillary beds drain into the hepatic portal vein, which instead of carrying the blood directly to the heart, instead carries it to capillary beds in the liver, which would be those sinusoid capillaries in the liver lobules that we had mentioned before. So all that blood and all the nutrients and toxins that it's carrying are going to pass through the liver, through those sinusoid capillaries, and the hepatocytes will have the opportunity to use the nutrients and to detoxify any toxins that are there. Then the blood will drain into the central vein of each liver lobule, ultimately into the hepatic vein and the vena cava. All right, so here you have capillaries, or I guess venules. It's not showing actual capillaries, so venules emerging from the large intestine, the small intestine, stomach, spleen, pancreas. All these venules are draining into veins, ultimately into the hepatic portal vein, a large vein that travels into the liver. Then it's going to start splitting into the portal venules. Then you have the blood traveling through the sinusoids of the liver, draining into the central veins, ultimately into the hepatic vein and the inferior vena cava. So because of the hepatic portal vein, you have the first pass metabolism, where any substance that was absorbed from your gut is going to be metabolized by the liver, or at least can be metabolized by the liver before going to any other part of the body. So that includes, of course, toxins. The liver will detoxify them, and it also includes drugs. So the liver will metabolize those drugs, sometimes activating them, sometimes just degrading them. And a prodrug, again, is a drug that is activated by the first pass metabolism in the liver. So that means that the CYP enzymes in the liver would actually be acting on that drug to make it active. The drug itself has no effect on the body, but the activated drug that's been metabolized by the CYP enzymes does have an effect on the body. So that's a prodrug. Other drugs that are very extensively degraded by the first pass metabolism, you might need to take it by a route that avoids the first pass metabolism, which usually those routes are a bit more dangerous, since your body's designed, or I guess the digestive system is specifically designed to take things in, to ingest things from the outside world, so that that's the safest way to ingest something. To get something into your body by a different route is always going to involve more risk. So the way to kind of avoid the first pass metabolism, but also take advantage of the fact that your digestive system is designed to ingest things, would be to, put it up the other end, either as a suppository or an enema, because the rectum actually of the large intestine does not drain, or I guess the capillaries in the rectum do not drain into the hepatic portal vein. The venules draining into the hepatic portal vein emerge, from the entire colon. Well, yeah, I guess actually the transverse colon as well, but not from the rectum. And finally, we'll talk briefly about how nutrients are actually digested and absorbed by the different parts of the digestive system. So first, an overview of digestion and absorption. Digestion has its two types, the mechanical digestion and chemical digestion. Mechanical digestion includes mastication, which is the fancy word for chewing, churning that occurs in the stomach, segmentation that occurs in the small intestine. The segmentation that occurs in the large intestine is not really for mechanical digestion, so that doesn't count as part of mechanical digestion. So mechanical digestion is occurring in the mouth, in the stomach, and in the small intestine. No matter where it's happening, it works kind of the same for different types of nutrients. There's not too much distinction between the types of nutrients as far as mechanical digestion goes, and you always have these larger pieces of food being broken into smaller and smaller pieces of food. But in mechanical digestion, what you do not have is chemical bonds actually being broken. So you're physically pulling molecules apart, but you are not breaking those molecules down chemically. You're not altering the chemical bonds of the molecules in mechanical digestion. If you are altering the chemical bonds, that is chemical digestion, not mechanical. The bile salts actually do count as a part of mechanical digestion as well, when they emulsify lipids in the chyme. Because they are, taking large fat globules and breaking them into smaller fat globules. They don't affect any of the bonds in those fats. They just affect how many fat molecules are kind of stuck together to form a droplet or a glob. Then for chemical digestion, sorry about that. For chemical digestion, it's mostly happening in the stomach and the small intestine. You also have some chemical digestion occurring in the mouth, but actually the bulk of it is in the small intestine. So you have a little bit happening in the mouth, some happening in the stomach, and a lot happening in the small intestine. In chemical digestion, you are breaking bonds between atoms in these molecules. So the nutrients, the major nutrients that you're taking in from your food are all called macronutrients because you need a lot of them, but they're also macromolecules because they are really big molecules. That would be the carbohydrates, the proteins, the fats. Those all form really large molecules, or at least can form really large molecules, and usually do. So in order to actually digest those things, you can't take them into the cells and put them in the blood when they're like super big like that. You have to break them down and make them small. So in chemical digestion, that's what you're doing. You're breaking the bonds of those macronutrients to get smaller and smaller pieces of them, and eventually individual building blocks, individual sugars, individual amino acids, or individual free fatty acids for fats. And you have different enzymes that carry out those processes. For each type of macronutrient, you're going to have a different set of enzymes that is responsible for chemically digesting it. Then finally, absorption. Most absorption occurs in the small intestine, especially for nutrients. You do have some absorption in the large intestine, mostly of water and ions though. Most nutrients would be absorbed in the small intestine. In absorption of nutrients, you have the building blocks of those nutrients, amino acids, sugars, free fatty acids, being transported across the epithelium of the small intestinal wall through the enterocytes that line it into the blood or into the chyle for lipids. So most of those nutrients will end up in the blood, only lipids will end up in the chyle in a lacteal. And again, that process is going to depend on what type of nutrient we're talking about. Each nutrient has a kind of a different path that it takes through enterocytes to get to its ultimate destination in the blood or chyle. So we'll start by going over the digestion and absorption of carbohydrates. Most of the digestion and absorption of carbs is occurring in the small intestine. Chemical digestion of carbs does actually begin in the mouth because in the saliva you have salivary amylase that breaks down large carbohydrates into smaller bits. But the food just isn't in the mouth for very long usually, which means that salivary amylase doesn't have much of a chance to get very far in that digestion process. So only maybe 10-15% of the carbohydrates are going to be digested by salivary amylase in the mouth. The rest of them would be digested in the small intestine. And you have several different enzymes that are responsible for that. You have pancreatic amylase, which comes from the pancreas, of course, that is going to break down your large carbohydrates that make it to the small intestine. They would break it down into kind of smaller pieces that would be called like oligosaccharides because they have just several sugars stuck together in a chain. Then you have a host of other enzymes, including lactase, maltase, and sucrase, that take those littler oligosaccharide chunks of carbs and break them down into individual sugars. Then it's the individual sugars that would actually be absorbed by the enterocytes. The exact way that they're absorbed depends in part on which sugar it is specifically. But most of those sugars can be absorbed by the sodium glucose cotransporter. Which is a type of secondary active transport, which is also called cotransport. So we went over cotransport in general biology, but in cotransport, it's a type of active transport, but you don't directly use ATP. So in active transport, you're always using ATP to pump some substance up against a concentration gradient. But in cotransport, you aren't using ATP directly. Instead, you're going to use ATP indirectly to establish a concentration gradient for something, in this case for sodium. Then your sodium glucose cotransporter will allow sodium to flow down its concentration gradient and use the energy of its movement to pump glucose or a different sugar up its concentration gradient. And it's, of course, the sodium-potassium pump that is going to be using ATP to establish that sodium concentration gradient, where you have a lot of sodium outside the cell and only a little bit inside the cell. So this diagram is kind of summarizing that process. You have polysaccharides that arrive in the small intestine that didn't get digested by salivary amylase because there wasn't enough time. You're going to have pancreatic amylase breaking these polysaccharides down into smaller oligosaccharides. This polysaccharide that's shown is actually already really small. Most polysaccharides would have way more sugars in it than that. But these oligosaccharides would just have a few. And then you're going to have the lactase, the maltase, the sucrase breaking these oligosaccharides apart into individual sugar molecules. Some of them might be glucose, some of them might be galactose, some of them might be fructose, and of course there can be other ones as well. Glucose and galactose would both be transported into enterocytes using the sodium glucose cotransporter. Sodium is flowing down its concentration gradient, and the energy of its movement is used to pump the sugar up its concentration gradient into the cell. That sodium gradient is established by the sodium-potassium pump, which is using ATP to pump sodium out and potassium into the cell. Then some sugars like fructose would be absorbed into the enterocytes by a different pathway using different channels to get into the enterocyte. No matter how the sugars get into the enterocyte, they get out through these transporters by passive, or I should say facilitated diffusion, so just traveling down their concentration gradient and then are absorbed into the blood. So similar to carbs, proteins are also mostly digested and absorbed in the small intestine. The digestion of proteins does begin in the stomach because you have the enzyme pepsin that's released by the gastric glands as part of the gastric juice and is activated by the stomach acid. So, and of course you have the stomach acid itself denaturing the proteins in the stomach. So between the proteins being denatured in the stomach and the action of pepsin, you have about 15% of the proteins digested into peptides in the stomach. But they wouldn't be broken down into actual free amino acids at that point. They would just be broken down into kind of small chunks of amino acids called peptides. Then once the chyme enters the small intestine, you have more enzymes released. The pancreas releases 5 different enzymes that break down proteins. One of them is trypsin. So those are going to take whatever proteins are left intact and break them into peptides. And then you also have a whole host of enzymes that come from the enterocytes themselves that finish breaking down those peptides into actual free amino acids. Then the amino acids and also really small peptides consisting of, just a couple amino acids will be transported into the enterocytes. You have a whole range of co-transporters that will do that, so it depends on the amino acid, like which specific transporter would carry it. But they're all co-transporters, so they're all consuming ATP indirectly. A lot of them are sodium co-transporters, so you would have the sodium-potassium pump using ATP to set up a sodium gradient where you have a lot of sodium outside the cell, only a little bit inside the cell. And then sodium co-transporters can allow sodium to flow into the cell down its concentration gradient and use the energy of its movement to pump an amino acid into the cell as well, up its concentration gradient. Then, as with the sugars, the amino acids would cross the other surface of the enterocytes, the basal surface, by facilitated diffusion, just going down their concentration gradient through a transporter and then ending up in the blood. So this diagram is kind of showing that most of that process, it's actually starting with a peptide already here. So in order to get your proteins to this state, they need to be denatured by stomach acid in the stomach. Then pepsin in the stomach will degrade some of those proteins into peptides like this, and the rest of the proteins will be degraded into peptides in the small intestine by trypsin or by other enzymes from the pancreas. Then once you have all the proteins degraded into peptides, then you have a bunch of different enzymes from the enterocytes that are going to chop that up the rest of the way into free amino acids. Those will be transported into enterocytes by some type of co-transporter. This is shown as a sodium co-transporter, and so you also have the sodium-potassium pump down here using ATP to establish the sodium gradient. Then the amino acids will cross the enterocyte and come out the other side by facilitated diffusion through some transporter. And finally, we'll talk about the digestion of lipids, specifically fats. So there's a number of types of lipids. Fats are just one type. Each type of lipid is going to be digested and absorbed in a different way. We're just going to talk about fats because that's the most common type of lipid that you would be eating. The process of digesting and absorbing fats is a lot more complicated than the process is for carbs or for proteins. So we're going to spend just a little more time on it. Like carbs and proteins, fats are mainly digested and absorbed in the small intestine. But like proteins, the digestion does begin in the stomach. So in the stomach, you have an enzyme called gastric lipase that is able to peel a fatty acid off of the fats, which gives you diglycerides and fatty acids. So actually, first let's go back and review the structure of fats. So a triglyceride here, this is a fat. A triglyceride is a glycerol molecule, which is shown in red, with 3 fatty acids attached to it, which is shown in blue. A fatty acid is just a chain of carbon and hydrogen with a carboxyl group on it somewhere. So these are all fatty acids. This would be a free fatty acid, and here's a free glycerol molecule. So if you take a glycerol and stick three fatty acids onto it, you get a fat, also called a triglyceride. So if you've ever seen like the word triglyceride on a product or something, that just means fat. So in the stomach, you have the enzyme gastric lipase that's going to snap one of these fatty acids off of the fat, and that leaves you with a diglyceride, a glycerol with two fatty acids attached to it, and a free fatty acid. About 15% of the fats in food would be digested that way in the stomach. Then everything will enter the small intestine and be digested the rest of the way and absorbed. First, in the small intestine, all of the fats and other lipids are going to be emulsified by the bile salts. So you're going to have bile salts kind of surrounding lipid globules and beginning to break them up into smaller and smaller globules or droplets. And each droplet will end up surrounded by a sphere of bile salts. It's important for the bile salts to do that because otherwise you have these really large lipid globules and it's just not practical to digest all of that. You have to break it into smaller bits so that the enzymes can access it and digest it. So once the bile salts have broken up the fats into little droplets and emulsified them, then you can have other enzymes taking effect. So you have the enzyme pancreatic lipase, which of course comes from the pancreas, that will further break down those fats and the diglycerides. So any diglycerides that were digested partially in the stomach already and fats that still remain intact, it's going to remove the fatty acids from the glycerol, all except for one of them, which gives you a monoglyceride and two fatty acids. So after pancreatic lipase has had its effect, you would have nothing left but just free fatty acids and monoglycerides, which would be a fatty acid attached to a glycerol. Those are still going to be associated with the bile salts, and the sphere that the bile salts form around each fatty acid or monoglyceride or around the lipid globules actually is called a micelle. A micelle is a structure that's formed by amphiphilic compounds in water spontaneously. So phospholipids will form it, bile salts will form it, soap will form it. It's essentially like a soap bubble. It's a little sphere made out of this amphiphilic compound where the hydrophobic parts always point to the inside of the sphere and the hydrophilic parts point to the outside. So in this diagram, it's kind of showing all those stages. So in the stomach, you have still large globules of lipids, of fats. They are going to be broken up somewhat by the churning of the stomach, and then you're going to have the enzyme gastric lipase come and break some of those fats into diglycerides and free fatty acids. So these little purple pill things are the glycerol, and the little squiggles coming off are the fatty acids. So that's a diglyceride and a free fatty acid. Then in the small intestine, you still have these lipid droplets. In order to fully access the interior of those droplets, you have to break them up further. So bile salts will do that by emulsifying the droplets until they're really small. Then you have pancreatic lipase come and catalyze the breakdown of those fats, giving you just free fatty acids and monoglycerides, a glycerol attached to one fatty acid. And then these fatty acids and monoglycerides will still be associated with the bile salts in micelles, like so. So you would have the bile salts all around the outside, and then on the inside you have kind of a little collection of several free fatty acids and or monoglycerides. Now you're ready for absorption. The absorption of fats is also kind of complicated. So first we're starting with these micelles of bile salts that contain a few free fatty acids and or monoglycerides. These things are really small, well, pretty small, I should say. So they're actually going to be able to diffuse across the membrane of the enterocytes, across the apical membrane. Because they are pretty hydrophobic, or they have a lot of hydrophobic parts, they are able to diffuse across that membrane. But in the process, they also break apart. So the bile salts themselves have their hydrophilic portion, they can't fully diffuse across the membrane because of that hydrophilic part that they have. So the bile salts are going to be left behind in the lumen of the small intestine, and only the free fatty acids or monoglycerides will actually completely diffuse across the cell membrane and enter the enterocyte. And then once you've got those free fatty acids and monoglycerides inside the enterocyte, they get reassembled right back into fats. So all that work you did to break down the fats into free fatty acids and monoglycerides, it was just to get that stuff transported into the enterocyte. Then as soon as it's inside, you put it back together as a fat. Then inside the enterocytes, you're going to assemble those fats into structures called chylomicrons, which are basically specialized micelles. They're made out of phospholipids instead of bile salts. And they're pretty large, larger than the bile salt micelles, at least. So you have phospholipids, a single layer of phospholipids in a ball around the outside of this chylomicron, oriented so that their hydrophilic heads point out towards the cytoplasm and their hydrophobic tails point in towards the fats that are inside of the chylomicron. Then the last component you have is the apoproteins. An apoprotein is just a protein that is able to bind a lipid. Then once the apoproteins have actually bound lipids that are in this chylomicron, then we call them apolipoproteins. So this diagram here is showing a chylomicron, or a section of a chylomicron. Around the outside of it, you have a shell of a single layer of phospholipids. Inside of it, you have a bunch of fats that is carrying. And then these blue bits are the apolipoproteins. They're called apolipoproteins because once the chylomicron is actually assembled, they're going to bind to lipids. And so it's actually this structure, the chylomicron, that is going to leave the other side of the enterocyte. And since it's so big, it cannot diffuse across the membrane. It has to leave by exocytosis. So the phospholipids are essentially going to act as a vesicle, and it will kind of fuse with the cell membrane on the basal surface and then exit. And then on the other side, instead of entering a blood vessel, since it's so large, it's going to enter a lacteal, which is that specialized type of lymphatic vessel that has really big flaps so that the chylomicrons can get through. So that means that fats are actually not subject to the first pass metabolism. All the nutrients absorbed in the small intestine that go into the blood go to the liver in the hepatic portal vein and undergo first pass metabolism. But fats do not go into the blood, they go into the lacteals to make chyle. Lymph mixed with fats would be chyle. So that's going to actually ultimately drain back into the blood at the inferior vena cava and not into the portal vein, so it doesn't go to the liver for first pass metabolism. And this diagram is just showing that process where you start with micelles that are filled with free fatty acids and monoglycerides. Those are going to diffuse across the cell membrane, but actually the bile salts cannot diffuse across the membrane, so those get left behind, and just the free fatty acids and monoglycerides end up completely diffusing into the enterocytes. Then inside the enterocytes, the cell is going to have enzymes that stick some of these free fatty acids back onto the monoglyceride, creating a triglyceride or a fat. Then those fats are going to be packaged into chylomicrons with phospholipids and apoproteins. Then the chylomicrons will leave the cell on the basal surface by exocytosis and enter a lacteal lymphatic vessel. And once you have those chylomicrons in the lymph of the lacteal, now it's called chyle.